everyone, today we, Bani and Suraya, will be reviewing a research paper titled The Optimization of Immobilized Lactobacillus pentosus Cell Fermentation for Lactic Acid Production. So, are you excited with this paper review session? If yes, then we shall start with the introduction part.
Lactic acid is known as hygroscopic non-toxic organic compound, which is massively used in various industries such as cosmetic, pharmaceutical, food, and chemical production. Then, polylactic acid have been developed and applied in many industries due to its benefits toward the environment, giving environmental protection and energy saving to the user. Polylactic acid have been the alternative choice to replace plastic production by petrochemical industry. There are two methods used in the production of lactic acid, which are chemical synthesis and fermentation. In chemical synthesis, there are some increment in the separation and purification costs. Not only that, this method requires a very high energy consumption. Compared to chemical synthesis, pure lactic acid can be produced using a specific lactic acid bacteria in fermentation method. Fermentation, lactic acid bacteria have shortened the time period for fermentation and decrease the costs for recycle process. Nowadays, method known as immobilized cell fermentation have been introduced and it carries many advantages. This method able to produce higher cell density in the fermenter, higher yield and production rate of fermentation, maintain high biological activity for long-term fermentation, and have a convenient product recovery. Immobilized cell can be separated from the fermentation medium and be used in repeated batch fermentation. This can reduce the time and the cost for cell culture.
The objective of this research is to optimize the concentration for lactic acid production by the fermentation of immobilized Lactobacillus pentosus ATCC 8041 cell fermentation. Now we shall move to the methodology part. In preliminary experiments, the factors that are required for fermentation of immobilized Lactobacillus pentosus are recognized based on the Blackett-Burman design. In this study, only P diameter, pH, initial glucose concentration, and biomass were discussed. The remaining two factors, the concentration of two colloidal solution, SA and PVA, with two cross-linking agent solution, calcium chloride and boric acid, and the temperature were already being discussed in the previous study. In seed culture preparation, the Lactobacillus pentosus strain must be stored in refrigerator at -8°C. The lyophilized cells then will be activated in D. Monroe-Gosa and Sharp MRS medium at 37°C for 8 hours on rotary shaker with 150 rpm speed. In cell immobilization, 2% SA and 6% PVA was added to the deionized water and undergo continuous agitation at 30°C and 8°C respectively, and was mixed subsequently to form SA-PVA hydrogel. Then, 100 ml of sterilized SA-PVA hydrogel was added with specific amount of L-pentosus strain. The SA-PVA hydrogel with the L-pentosus strain earlier was then injected into mixed cross-linked agent consists of 0.1 molar calcium chloride and 2.5% boric acid using electrostatic droplet generator. Then, the immobilized cell beads were prepared with a certain diameter in sphere sheet. The immobilized cell beads were then kept in mixed cross-linking solution and stored at 4°C for 4 hours. The beads were subsequently washed by sterilized deionized water to remove the calcium chloride and boric acid. In batch fermentation, the working volume was 800 ml and fermented in 1 liter of new Brunswick bioreactor bioflow for 84 hours. The impeller was removed to avoid any bead damage. The agitation was done at 150 rpm speed by using magnetic stirrer. The recipes for this medium are shown in the video, which include 5 g per liter of peptone, 5 g per liter of yeast, 0.5 g per liter of potassium dihydrogen phosphate, and 5 mol per liter of sodium hydroxide. The temperature was controlled during this fermentation. For chemical analysis, the concentration of glucose and La were measured by protonuclear magnetic resonance spectroscopy (NMR). The NMR sample include 0.5 ml fermentation sample, 0.4 ml deuterium oxide, 0.1 ml internal standard which contain 95.5 Wh% deuterium oxide, 4.2 Wh% glucose amines, 0.2 Wh% trimethylamine, and 0.1 Wh% trimethylsilyl propionate. The peak was interpreted using Mass Renova software at 5.25 ppm, 4.65 ppm, and 1.35 ppm. The curve obtained show the relationship between the concentration and the peak, which are the concentration of glucose and La. For statistical analysis, the researcher used the formula in the video and calculate the ANOVA. Let's see the result part. For chemical analysis, Table 1 shows the experimental values as format mean value plus minus standard deviation. The analysis of variance, which is ANOVA, was carried out by design expert version 11. The response surface was applied to describe the effects of different factors on the efficiency of immobilized L-pentosus ATCC 8041 cell fermentation. The quadratic models of LA yield and LA productivity were generated to show the functional relationship between factors and responses. The statistical analysis based on Table 2 and Table 3 shows the ANOVA of the quadratic model that were used, and they confirm that the model is adequate and reliable, which could be used further to navigate the design space. These two figures show the effect of the parameters on the immobilized L-pentosus ATCC 8041 cell fermentation. Based on the figure, the LA yield and production increase with a decrease of bit diameter, which indicate the promotion of mass transfer performance and reduction of product inhibition by smaller bit diameter. As the bit diameter decreased, the surface to volume ratio of the bits increased. This led to a decrease in diffusion resistance, hence the substrate can easily diffuse into the smaller size bits and be utilized by cell, which resulted in an increase in product yield and productivity.
Let's discuss. Lactic acid yield decreased when the bit's diameter was equal or larger than 5 mm. It can be concluded that biodegradation efficiency increased with the decrease of bit diameter from 4 mm to 1 mm. Substrate with higher concentration can diffuse easily in bits with larger diameter. It was found that the highest lactic acid production is at pH 5.5. Higher pH inhibited the activity of ATPase combined with a change in pH of cytoplasm, which decreased the fermentation efficiency. Hydrogen ion in the fermentation diffused in the large pores of calcium alginate matrix and caused changes in charge quantities of plasma membrane. This will change the permeability of plasma membrane to nutrients and inorganic salts ion. Extreme acidic condition can cause changes in the intracellular ionic environment and damage the protein structure, which decrease the production yield and productivity. If the pH is too low, it can also inhibit the reaction of enzyme. When the concentration of glucose is 80.6% or higher, it will lead to a significant restriction of lactic acid production. This is because the concentration of glucose will affect the ethanol production of immobilized bacteria in batch fermentation. Higher glucose concentration also causes higher retention time for higher yield due to the low feed dilution. Feed dilution is the one causing the cell osmotic effect on the mass transfer performance and cell activity. High glucose concentration also reduces the water activity and plasmolysis, which decrease the fermentation efficiency. Although, if the glucose level is too low, the production yield will also decrease because there is not enough nutrition for the cell to grow and develop. For biomass, it is quite simple because the production of lactic acid will increase when there is a higher amount of biomass provided.
In conclusion, the immobilized L-pentosus cells perform with a high fermentation efficiency and excellent stability in homofermentation for LA production. The parameters have been optimized to be 2.0 mm bit diameter, 5.60 pH, 115.3 gram per liter initial glucose concentration, and 398.2 mg biomass CDW based on response surface methodology with box packer design for maximizing the LA yield. The highest LA yield and productivity have been obtained as 0.938 plus minus 0.03 gram per gram glucose and 2.213 plus minus 0.008 gram per lbs respectively. The stable and high LA yield and productivity can be obtained by repeated batch fermentation by pentosus cells under optimum conditions.
Well, that's all from us. Thank you for watching.